Hi all, Dr. Clark here again, and today we're going to finish up kind of talking about mitosis. We'll move into meiosis, which you're going to see there's a lot of similarities between the two. But today we're going to talk about cell death, or more specifically, in many cases, we're going to talk about apoptosis, which is programmed cell death. And so here's kind of a picture of programmed cell death. So you have this cell and something's wrong with it. Maybe the DNA inside is damaged or something's going on. And the cell itself triggers basically suicide. And so it goes through this process of digesting itself from the inside out. And so the lysosomes would erupt and you get digestive enzymes that are made in masses, and it would just start digesting everything in the cell. And eventually it'd just be particles that could be then picked up by other cells and utilized by other cells. Now we do this often on little pieces, like, maybe you got a mitochondria that's damaged, or your Golgi apparatus might be damaged or something in a cell. So you'll have a lysosome will kind of engulf that or destroy that, and then you'll make a new one, or new ones if it's multiple. But apoptosis is the entire cell, right? Or in some cases, all the cells in a given region. Okay? And that's what we're going to look at a little bit today. So cell death, this happens a ton when we're developing in the womb, and well, for that matter, all organisms that are developing will have some form of cell death. So from a human perspective, though, there's lots of ways that we can point to regions of our body that went through apoptosis, a controlled cell death, or programmed cell death. So, I mean, one that's probably, the best example that I can give that you can, distinguishingly look at, you can see videos, you can see photographs, and that is that when we're in the womb, we have skin flaps between our fingers and between our toes. And as that individual, as that fetus starts to develop, apoptosis happens, and the fingers get split. So the skin flaps that occur between have programmed cell death, and the cells die back. Now, occasionally you will have a child that's born with skin flaps between his fingers or her fingers, and doctors just clip it. Okay? For example, on a personal note, my daughter was born what's called tongue-tied. And so you have a little teeny flap of skin that holds your tongue to the bottom of your mouth, and that goes all the way out to the tip of the tongue while you're developing. But through apoptosis, it recedes, so the tongue can be released. Okay? But in her case, it didn't recede enough, and so her tongue barely could move. And so all the doctors do is they just pick up the tongue and they clip it and they cut it. And when you do that at a really young age, it's not going to grow back. The clip stays, and then the tongue is released and it's free. So this is just a case where, genetically speaking, from at least my daughter's point of view, she didn't have the right genetics to release her tongue. Just like some children will be born with, skin flaps between their fingers or between their toes, or an extra skin flap here or there, because cell death or apoptosis didn't happen completely. So that occurs, and that's occurred genetically, and it's triggered by, well, in many cases it might be triggered by the turning on or off of other genes. For some regions, we don't really know what's triggering why it occurs at a certain time. For other things, we know exactly what gene was turned off or turned on, what gene has been manipulated, and that can induce that apoptosis. But in other cases, maybe we're not interested in destroying cells per se, but deciding whether or not a cell can go through cell replication. And that's the cell cycle. We've already talked about the cell cycle. So remember, we talked about interphase, which consists of the G1, the S, the G2 phase. Then the M phase, which is either for mitosis or meiosis. And then the C phase, which is for cytokinesis. And that makes up the cell cycle. Now, cells happen to get damaged all the time. Cells are old, and so they can age. And old cells aren't as functional as young cells, and so a lot of situations go where you need, or your body needs, to control the cell cycle. You need to be able to control what cell is allowed to reproduce and what cell is not. And that feedback will allow for a cell to either progress to the next portion, the next stage of the cell cycle, or not progress. And so there's a few checkpoints, but the three main ones that we're going to talk about, and probably the ones that are best known, are a checkpoint that occurs at the end of G1, which is called the G1 checkpoint, a checkpoint that occurs during G2 checkpoint, and then a checkpoint that occurs during the M phase. These are probably the three most documented and most well-known checkpoints, and are the ones that most of what we know issues with, like uncontrolled cell growth, which is what cancer is, involves one of these three checkpoints. Okay, so checkpoint 1, again, like I said, it occurs right before a cell is going to enter the S phase, or the synthesis phase. And what's happening is you have a series of proteins that ensure that things like your DNA is not damaged. So you have a protein called P53, or protein 53, and then you have another protein that's called P21, and you have a whole series of proteins that the role in the body is to check your DNA for mistakes. And if there is mistakes, and they're simple mistakes, like maybe a wrong base pairing, so maybe adenine's paired to guanine, then it can just reverse that. It can remove the guanine, add a thymine, and it's not a huge deal. But in other situations, maybe the DNA is damaged so much that there's no fix to it, and that cell needs to be destroyed. So there are proteins that do this, that make those checkpoints. They're proofreading proteins. Now, it's not just about DNA though. Right? It also occurs, well, is the cell big enough? Right? Is the cell, does the cell have enough mitochondria? I mean, is the cell's cytoplasm large enough for it to go through this phase? And if it's not, then often these cells will either go through programmed cell death, or they'll be sent to what we call the GO phase, or a phase that's outside of the cell cycle, and the cell can never pass through the cell cycle and never complete the cell cycle again. That's what happened with your nerve cells. Okay? So, you might hear these terms, well, don't destroy your nerve cells because you'll never replenish them. They don't grow back. And to a point, that's true. You have stem cells in your body that are undifferentiated cells. And so they're just not determined what kind of cell it's going to be yet. So you can have small damages to nerves, and stem cells can then be turned into nerve cells, or that could be their function as a nerve cell. And so you could rebuild synapse and rebuild nervous tissue in certain regions. But overall, it is correct to say that nerve cells do not go through the cell cycle. And what they're going through is they're violating the G1 checkpoint. So for some reason, some evolutionary quirk, the GO phase is where the nerve cells go from day one. So nerve cell differentiated, if it tries to go through the cell cycle, it's pushed to the GO phase. And GO phase, for lack of a better term, is kind of like the old person home. They're still in existence. They can still function. In the case of nerve cells, they function perfectly fine. But in the case of other cells, they can still function. You're just not allowing them to reproduce. You're not allowing them to, produce a bunch more of them because there's something wrong, or in some cases, there's some quirk of evolution that pushes them into the GO phase. The G2 checkpoint, that's what's going to lead into mitosis, or in some cases meiosis. And that G2 checkpoint is going to ensure that after the S phase, that the DNA has copied correctly, that you don't have huge mutations. It also ensures that you do have enough mitochondria. You have meiotic spindles that formed. You have enough cytoplasm. The cell is big enough that it can go into mitosis. And then the M checkpoint is, the purpose of this is to ensure that during metaphase, when all the chromosomes are lined up on the metaphase plate, that they're all connected to a microtubule. Not to one, but to two microtubules. So each sister chromatid is connected to a microtubule. So when metaphase moves into anaphase, that those chromatids, sister chromatids, separate. Because if they're not connected, so maybe only on one side it's connected, the pair of sister chromatids will move to one cell. And you'll have twice the number of chromosomes in one cell than none in the other cell. Now this happens, and we'll talk about this from a sexual point of view. When we talk about meiosis, we'll talk about different sexual, sometimes called sexual disorders, but sexual mutations that occur from non-disjunct chromosome separation. So you just get chromosomes that move to one cell, and one cell doesn't have that sex chromosome, and the other cell does have that sex chromosome, or both sex chromosomes. Okay, and we'll look at this and we'll talk about it more. But that's the purpose of that M checkpoint, is to ensure that they're all connected and the metaphase can move into anaphase. So here they are. So this, you can just think of the cell cycle as a cycle, because that's what it is. And remember that G1 phase is the normal kind of stage that most of your cells, if they're not going through reproduction, are sitting at. So the normal function of the cell will be sitting in G1. But then, let's say you get a cut, or you need a new cell lining in your stomach, intestines, you're building muscle, something, you need to add new cells to a certain region. And the cells that are nearby that region are going to try to go through the cell cycle. So they're going to be triggered to go into replication mode. They first have to make it past that G1 checkpoint, and this is the main checkpoint that is violated when things like cancer occur. And this would be a violation of the p53 protein, okay, typically. So that G1 checkpoint might be violated. Then you copy whatever mutations you have, if that's the case. Now sometimes the G2 checkpoint will pick up those mutations and consider that cell a faulty cell and not allow it to pass. Other times it passes and makes it into the M phase, and as long as all those chromosomes are picked up from microtubules, or chromatids are picked up by microtubules, it'll pass the M checkpoint, and then it'll go through cytokinesis, and you just created mutated cells. We'll look at this in a second. Now, before we get to that though, the age-old question, or maybe it's not age-old, but I mean, humans have been around on the planet well, in our modern form, Homo sapiens, probably for close to 120,000 years. We've been out of Africa for maybe 40 to 50,000 years. There's been humans in North America probably for 15,000 years. So given that information, Homo sapiens, I don't want to say we're the smartest species on the planet, but there's a lot of things that separate us from other organisms, and intelligent capabilities is clearly one of them. So we do have high intelligent capabilities. Our actions may not be the smartest, but our capabilities are there. Okay? Now, forever, since we've known about life and aging and death, people have been trying to figure out, why do people die of old age? What is the limit? Why is 120 years, roughly 120 years, the maximum lifespan for humans? I think the oldest person ever recorded was 122. Okay, so that 120-year mark is pretty much it. Right? Why? Well, a guy named Leonard Hayflick in 1961 tried to answer this question. So he did this by demonstrating that fibroblast cells can divide about 50 times, and then they can't divide anymore. And the cell doesn't go through apoptosis, it doesn't die, but after 50 times, it can't pass that G1 checkpoint. And so it gets put into the GO phase. And you have this stationary cell that's sitting there and it does nothing. It can't replicate anymore. And Leonard Hayflick, really didn't have an idea of why this occurs. He just showed that it did occur. So it took a little bit of time, but in 1978, Elizabeth Blackburn won the Nobel Prize for an explanation for the Hayflick limit. And what she found out, that when you look at chromosomes, and I should have shown you this when I was talking about chromosomes, but we've already talked about chromosome structure a little bit. So in the center of the chromosome is what we call a centromere. But at the tips of each one of your chromosomes, at the ends, you have what we call a telomere cap. Okay? And normally this is just repeated adenines added to the end, or sometimes it's called a poly-A tail. Okay? And so it's a portion of your chromosome that doesn't code for anything. There's no genes in there, and it's a protective cap. Well, what happens is, through DNA replication, as you replicate your chromosomes, every time you replicate your chromosome, a little piece of that cap is not copied. Okay? And that's because we copy our DNA in two directions. One in what we call a leading strand, and it's continuous, and one that we call a lagging strand, which is discontinuous, and it's made of what we call Okazaki fragments. And we'll come to this when we talk about DNA replication. But that lagging strand, because you have to have a primer that lays down every time, the lagging strand always has just a little bit that is not copied, okay? Which results in your chromosomes over time shrinking. They get smaller, and the telomere cap is being removed. Well, eventually, when the cap is gone, the cell goes into the GO phase. It's violating it violates the checkpoint, and it's pushed into the GO phase. And so in humans, at least, it's about 50 replications. Okay? So about 50 replications, and our telomere cap is gone. Okay? So each one of your cells has about 50 replications. It just happens to be that based on the number of cells we have, starting out at, as a young individual at birth, we have a certain number of cells. Now they're going to replicate as we grow and things like that. But starting out at that size, and that about 120 years is the maximum. I mean, that it takes before enough of your cells are completely stationary in the GO phase that you can't fix things. You can't go through maintenance. You can't fix little blemishes or anything like that, and your body's giving out. Okay? Now, some people, it's faster because of maybe diet, because of genetics, because of lots of things. So some people, their life expectancy would never be 120. Other people, maybe it's in the hundreds, maybe 110, maybe 120. And that's, could be related to how much of the telomere cap is removed each time. We know things like Galapagos tortoises and, other organisms that live for a very, very long time. We know that when they divide, their cells divide, less of the telomere cap is removed at a time than, say, human cells. So they can actually replicate more times than we can. And organisms that don't live as long, maybe they have a life expectancy of like 10 years. So maybe you're talking maybe like a mouse, maybe a life expectancy of 5, 10 years. It depends on the species of mouse. Their telomere cap gets removed faster, and so they have less of a life expectancy. And so again, these cells, after about 50 replications in humans, they'll go into, a period of no reproduction. Okay? And this is why things, start drooping, and you get, crow's feet and wrinkles, and your hair turns gray and things like that, because you still have the cells there. There are still cells there. They're just not reproducing. And so for a long time, since really 1978, there have been researchers all over the world trying to get the Holy Grail, trying to get eternity life or extend our life expectancy by doing different things. Now we know in reproductive cells, okay, so in germline cells, we know that there's something called telomerase, okay, and it's an enzyme that adds new telomer caps back on. So telomerase adds new telomere caps back to the ends of your chromosomes in germline cells. So this is why, like, males can reproduce, and can produce sperm basically constantly until they're, 60, 70 years old. I mean, depends on the individual. But for a long time, you're reproducing millions of sperm constantly. And it has to do with the germline cells. They can reproduce a lot faster and a lot more, right? Because that telomere cap gets added back on. It just keeps getting added back on. A protective cap gets added on from telomerase. So there have been lots of researchers all over the world trying to figure out, well, is there a way that we can add telomerase to somatic cells, to all the cells except, besides your germline cells? Is there a way that we can do this? And that would extend our life expectancy, extend how long we live. Okay? Now, that's one approach. There's another approach out of some researchers in Japan, which instead of extending the life of a given cell, if you can remove the cells that are in the GO phase, then you don't get, in certain regions, you wouldn't get a buildup of all these cells that can't do the cell cycle. So instead of having a bunch of GO phase cells that are stationary and census, and not doing their normal job but not reproducing, if you could remove those and have new cells come in that are younger and have, another 25, 35, 45 replications in them, and you keep doing that, then you could extend the life. And it looks like they've got a little bit of support with extending the life expectancy of mice and things like that. But I'm just saying that there is definitely a lot of research on this, because this is the way to extend human life. This is the scientific holy grail. And there's been lots of books and things written about it. Both these books I highly recommend if you're interested in how to extend life and things like that of humans. I'm not saying that, you know, these things work. I mean, if they tell you to change your diet, I'm not saying it's going to extend your life. But there's a lot of science behind it. This book is written by Elizabeth Blackburn, who won the Nobel Prize for, you know, the telomere effect, or the decreasing of telomere. And there are things that can affect your replication of cells and your replication of telomere, or how much telomere is removed each time. At least based on, you know, some scientific evidence, that's the case. And so she puts forth some, you know, nutritional facts and things like that that might help with that. The telomerase revolution really has to do with that enzyme telomerase that's added in your germline cells, and there are a few scientists that are working at trying to get it added to other somatic cells so that you can reduce the effects of aging, especially like Fossil, who is a well-known researcher of Alzheimer's, and has been trying to use this to reverse or to prevent Alzheimer's from happening in individuals. Great books. Check them out if you like. All right, so that kind of brings me to cancer. I mean, it's a bad word. About 1 in every 4 people will develop cancer in their lifetime. I think that's what it is at right now. When I graduated grad school in 2010, the report was 1 out of every 7. So in 8 years, either we've discovered new cancers, which that has happened, or the rate of people getting cancer has increased. That's probably also has happened. Now it's 1 out of every 4, and then if you increase the chances by doing things that are known carcinogens like smoking and things like that, it's about a 50% chance, maybe less, a little less than 50% chance, but you definitely increase the rate at which, or the chance that you would get cancerous cells. Now, cancer itself is a quite normal process, right? And cancer in general, probably every single person, you know, on the planet has probably had cancer before, or even has cancer in their body. Because cancer, the term, is really just based on growth, overgrowth of cells. So uncontrolled cell growth, at least initially, uncontrolled cell growth. And so what happens is the cells bypass the cell cycle and they just keep replicating. Now, what often will occur is that replication occurs really fast, and you'll form a tumor or a cluster of cells that just replicated in a certain region. So this is a lung tumor here, a lung cancer. And it's just cells that over-reproduce and just keep on reproducing and keep spreading. Okay? Now, cancer itself is uncontrolled cell growth. Cancer, when we talk about lay terms, when people say it's cancerous, okay, then it's saying that it's spreading to other parts of your body. Okay? And so this is a different type of cancer. All right? So there are 2 main types of cancer. You have benign cancer, which I say most people have had, okay, and have right now, is benign tumors, which is just, you know, your cells bypass the cell checkpoint for a little bit, and then they get encapsulated. So your body just encapsulates them and prevents them from spreading, prevents them from growing anymore, for that fact. Okay, and they don't spread to other areas. And that's a quite natural process in lots of organisms. I mean, almost every single organism has this process where they can encapsulate cells that are starting to form a tumor. And if they do that, then those encapsulated cells, they shouldn't spread to other areas, and the tumor is considered benign and pretty much harmless. Okay? The ones that most people are interested in and what we normally deem as cancer is malignant tumors, or tumors that are not encapsulated, because they tend to metastasize. And so they tend to break off small chunks of cells, and they will, those cells will move to a different region. Those cells are still going to bypass the cell checkpoint in that region, in many of the regions. They're still going to bypass it because the overarching problem has not been fixed. Okay? If cells are bypassing your G1 checkpoint, there's something wrong in the system. Your evaluator, your checkpoint evaluator, is broken, most likely. And so you're going to have these cells just keep replicating. And so when you have malignant tumors and they break off and they float around in the body, then you can get other tumors forming in other regions of the body. And this is where cancer, as we know it from a human standpoint, if someone has cancer, this is normally what we're talking about. Now, a lot of cancer, when we think about cancer in general, that inability to encapsulate or inability to stop the cell from bypassing the checkpoints is due to damaged genes. Okay? Now, it could be due to poor genetics. And we'll get into this in just a second. That's not saying that you can inherit cancer. I'm not saying you can inherit cancer. But you can inherit genes that are not as functional as someone else's genes in preventing cancer from forming or encapsulating or lots of different things. Okay? And so what ultimately happens is genes will be damaged, and then they can bypass the cell cycle, and then you get these damaged genes being replicated over and over and over again. Okay? And that's all a mutation is really just a change in the genetic code. Now, most mutations are either benign or have no real effect, or they are bad. So they are a dysfunction or a disorder, or they do damage to the individual's body. Now, there are ways at which mutations can happen. It can just be natural. Right? You're copying your DNA and your DNA just makes a mistake. Nothing happens, it just made a mistake. Now, when you copy 3 billion base pairs, 3 billion ATCs and Gs, you're expecting for at least a few mistakes to happen. I mean, that's a lot of code for your body to go through and make, without making mistakes. So you're expecting a few to actually occur. Now, there are ways at which we can increase the number of mutations, and that's by doing things like exposing ourselves to carcinogens. And one that we know a lot about is UV rays. Okay? UV radiation, especially in humans, can cause what we call thymine dimers. So exposure of DNA to UV radiation can cause thymine on the same side of your DNA, so on the same strand of DNA, to bind to itself. So thymine bound to another thymine, right, which will cause a little kink in your DNA. And then when it's replicated, sometimes that kink is recognized as just a single thymine, or the opposite side is recognized as just a single adenine. So you get a mutation and you're missing a base pair on either side. And that's called a thymine dimer. We've known about UV radiation and its effects for a long time. Right? That increases the rate of mutations. And if you increase the rate of mutations, you increase your chances of developing cancer. Now, there are lots of other things that we don't know as much about, like viruses. We know that there seems to be some links to cancer and virus. Like if you've had certain viral infections or been infected with certain viruses, you have an increased likelihood of developing certain types of cancer. Now that research is relatively young, but there's starting to be quite a bit of, quite a bit of scientific literature that's showing the link between viruses and DNA. Now, there are two general classes of genes that are involved with cancer. The first class is what we call proto-oconal genes. Okay? And these are genes that cause cells to divide. So basically what it's doing is proto-oconal genes are telling the cell to start dividing. So it's a sensory system where it's stimulating cell division. So it's stimulating a cell to go through the cell cycle. Well, in some cases, that is a mutation. And by doing that, the cell just keeps going through the cell cycle, and it keeps mutating over and over and over again. Okay? And we call this oconogenes, or oconogenesis. Now the other one is basically exact opposite. These would be tumor suppressor genes. Okay? So you have a series of genes that are inducing growth and inducing movement through the cell cycle. And then you have a series of genes that are trying to prevent uncontrolled cell growth. Okay? And when you have a mutation in either one, that cell cycle can be bypassed and it can have continuous cell growth. So tumor suppressor genes are, going to prevent cells that might not be healthy from going through the cell cycle. But if you have a broken tumor suppressor gene, then cells, no matter what they look like, however damaged they are, they can just keep moving through that cell cycle. It's called uncontrolled cell division. Now, a lot of cancers, right, and we're starting to learn a lot more about this as time goes on, a lot of cancers are the result of these damaged genes. Okay? And so it's the result of these failing cell divisions. Probably the one that's got the most evidence and the most support is a gene called P53, protein 53. Okay? This is the gene that its job is to check your DNA at the G1 checkpoint. And so if it detects abnormal DNA, it will either fix the DNA or trigger another series of enzymes to come in and fix the DNA, or it'll trigger some enzymes to come in and destroy the cell and go through apoptosis. Now here's the issue, and there's a lot of cancer, okay? So lots of different types of cancer. Well over 50% of cancers that we know are the result or have some process, okay, that is linked to the P53 gene. Okay? So if your P53 gene becomes damaged or is faulty, okay, then these damaged cells are unchecked at that G1 checkpoint. So then that means that damaged DNA can be copied during the synthesis phase and on and on and on. So here's kind of a diagram of how that works. So you have a P53 protein. Maybe you get some UV radiation that causes some thymine dimers, or you get exposed to something, some kind of carcinogen, causes damage in your DNA. Your P53 protein comes in, fixes it. Okay? Now, if it fixes it, you can have normal cells that are perfectly fine, okay, and can go and divide and go through the cell cycle, etc. But if it can't fix it, it's just not possible, then that cell is faulty and it's deemed and it's marked as a faulty cell, and it goes through apoptosis. So it's triggered to destroy itself. Now if you have an abnormal P53 protein, so this P53 protein is broken. Now remember, your proteins are genetically, or the codes for your proteins are genetically inherited. So you as an individual could inherit a faulty P53 protein. So the genetics of that could be faulty in the sense that you could have that region of your DNA could mutate faster. The protein might not be completely legit in the sense that it might not have folded quite correctly, and it just might not be very good. This is why doctors will often ask you, do you have a series or a history of breast cancer in your family? Do you have a history of colon cancer in your family? It's not because you inherited cancer from your mother or your grandmother or your grandfather. It's because you might have inherited a poor p53 protein system, or another checkpoint system might be faulty. And knowing that could benefit you in the future, because the sooner you get checked, the earlier it gets detected, the more likelihood that you would get or potentially receive treatment that would work. So let's say you do inherit a abnormal p53 protein because the genetics is poor. That means this doesn't work very well, right? Which means that when it tries to fix this, sometimes it might fix it, okay? And you might get a fixed chunk of DNA and you might get perfectly good cells. Other cases, though, it wouldn't fix it at all, and it still passes the G1 checkpoint. And some of these cells would become cancerous. And that's kind of how a lot of cancers form. All right, now so let's watch a video about how this is done, not just with p53, but some other known suppressor genes.
During the first phase of the cell cycle, G1, there is a chemical checkpoint that a cell must pass to begin the process that leads to cell division. The protein products of several tumor suppressor genes act at that checkpoint to control cell division. Cells with mutated forms of the tumor suppressor genes may not be able to stop the cell cycle at the G1 checkpoint, thus allowing them to grow and divide rapidly and form a tumor. One such tumor suppressor gene is the retinoblastoma, RB gene. It is often found to be mutated in a form of cancer in the eye called retinoblastoma, as well as in many other cancers. Normally, the RB protein will bind to a transcription factor called E2F. Transcription factors are proteins that can activate gene expression. The G1 checkpoint is passed when special cell cycle proteins cyclin and CDK work together to free E2F from RB. The released transcription factor then activates genes that start the cell on the path to cell division. Mutated forms of RB may not bind to E2F, allowing E2F to act without control to promote cell division. Another tumor suppressor protein that works at the G1 checkpoint is p53. This protein checks for damaged DNA, acting as a quality control for the cell. If damage is detected, then p53 both activates a DNA repair system and halts the cell cycle at the G1 checkpoint until the damage is repaired. The halt is accomplished by turning on the gene for another protein, p21, that keeps cyclin and CDK from interacting. As long as cyclin and CDK cannot form a complex, the cell cannot pass through the G1 checkpoint. The p53 gene is found to be mutated in a wide variety of cancers. In these cases, p53 fails to turn on p21 and thus does not halt the cell cycle like it should when DNA is damaged.
Okay, so there you can see some different examples, and I will tell you this, that we've known about this for a long time, and as much as you'll hear me rant and rave at times about pharmaceutical companies, and maybe they're not the most honest people because they're trying to make a lot of money. There are some new drugs out that are showing some promise. And the other thing about drugs and pharmaceuticals and things like that, it takes a very, very long time for that drug, even if we know about the p53 issue, we know about the p21 and these issues here. If we can devise a drug that could halt the checkpoint and not allow for that tumor to grow anymore without radiating it, and that's what chemo does. But if we could do that, then we could show a lot of promise with cancer treatments. Now, the issue often that we have is the process by which new drugs come about is a very, very long process, very expensive process. and there's a lot of regulations. So there are some drugs out there that show some real promise, and we'll probably talk about those later on in the semester. If not, you can Google them, P53 drugs or P21 drugs, and you can see that there are drugs out there that have been used for cancer treatments in mice and rats and things like that, that show some real promise, some real capabilities of decreasing tumor size and ultimately treating cancer. It's just that we still got a long ways to go. We really do. And again, like I said before, early detection is the key to prevention. And we'll talk more about cancer later.